childhood obesity epidemic is a real and consistent problem here in the United States. And what used to be just a concern for older, overweight people is really now becoming a pediatric concern. Any drug that's being given to patients in the United States is likely also being given to patients with the severe form of fatty liver disease. And so a legitimate question is, what does the status of their liver disease have to do with the ability of that patient to metabolize and eliminate drugs in a normal manner? These same drug metabolizing enzymes and transporters that determine the fate of drugs also determine the fate of environmental pollutants, toxicants that our bodies are exposed to all the time. And so the grant that we put in was geared toward adverse drug reactions, but they were really looking at the mechanisms by which our bodies interact with the environment, anything from the outside. And so we have extended that work not only with drugs, but also with environmental toxicants. The only way to determine whether you have this severe form of fatty liver disease is to do a liver biopsy, which has a lot of negative side effects associated with it. And so the idea of getting a non-invasive way to determine the stage of fatty liver disease is important. And so we were able to do that with these children. And so now, instead of having to have a big needle stuck into your gut and have a chunk of liver sucked out, you can essentially go in and take 2 Tylenol and then give either blood or urine. And based on the levels of this particular metabolite, we can get a good approximation of whether you have that severe form of fatty liver disease. And so we patented that idea and licensed that to Raptor Pharmaceuticals. So in order for us to use that as a diagnostic biomarker, we had to have a statistical comparison of its efficacy and so essentially its predictivity and its false predictivity to see whether it would work correctly. And so that's a statistical analysis that was done by Dean Billheimer. We have used his statistics to essentially state that our test is functional, and we used that in the grant that we just received this past year, and we'll continue to use that for another pediatric adverse drug reactions application that we're putting in next month. So we have had a couple of lab meetings where we discuss specifics about what we need before we can publish and what we can get from our clinical collaborators. And more patients is always better, but dealing with real-world limitations on the number of study subjects that we have available to us, they understand those limitations, and they can help me work around those limitations in order to get a reasonable publication and a reasonable grant application. And it has been my experience with those guys. Their written reports and the way that they come to an understanding of the questions that we're asking, they are very adept at understanding the questions asked, defining it in a statistical experimental outline, and then giving us analyses that are quality, that add value to what we've done, and allow us to move